Thank you. I'm Bill Heeschen. I'm from the Dow Chemical Company, and I'm a bit of a fish out of water. We do not do a whole lot of biology. We have a fair amount, but I'm going to talk a little bit about a little more of a material science type application. But the principles are all the same. Once you got the picture in, it's just a bunch of pixels, and you got to do something with it. In this particular case, we're looking at extruded polystyrene foam. This is the styrofoam that goes like on the wall of your house. For insulation purposes, or if you guys are craftsy people, sometimes you've arranged flowers by sticking the stems into pieces of foam. It's probably ours, or at least it's something like ours. So I'll start out by giving a little bit of a motivation on the background of the problems that we're trying to solve here. I'm going to spend a fair amount of time talking about the image analysis because that's what's of interest to this group. And then following up on an interesting aspect of something that Benjamin was kind of getting at. We rate the appearance of these foams by panel human rating right now. You just kind of look at it and say, is it good, is it bad? And in a second, you'll see what we're talking about. We end up often trying to solve the problem of having historic data based on human rankings, panel rankings that we then have to translate into numbers, and that way we can get an objective view and that sort of thing. Then talk a little bit about what we're going to do next. All right, so up here, there we go. This is what we want it to look like when we make this foam, nice and even, consistent appearance. But sometimes what will happen due to changes in processing conditions or changes in the formulation that we're doing, we'll get kind of a period of time when the foam comes out with a weird pattern in it. And by and large, the pattern doesn't really change the behavior of the foam, but it changes the way that our customers perceive the foam, and so we really don't like to have this pattern generally appearing. So, the pattern is a result of small and large cells mixed together. The sharpness and the extent and the strength of the pattern is based on how many of these cells are strung together at the small size, how many at the large size, how sharp are the boundaries between the small and the large cell domains. And how long do those domains last? So the problem of any of you who've been involved in panel ratings know that they're nasty. You have to have well-trained panelists who really are very diligent about ranking what they're looking at, and even then you don't generally get really good consistency out of them. We use this as part of our kind of our ongoing production system to watch how these foams are being made, and as long as they look like that one, we're happy. But they don't always. So we end up having to check the pattern. The first step in these, just for reference, the upper left corner, that piece of foam is about 1 foot by 2 feet or so. So our size domain is quite a bit different than where most of you typically work. But like I said, once they're pixels, they're just pixels. So the first step, and I'm not going to spend any time, we crop and we straighten the image, and we center the pattern and all these other good things. But then once we're ready to go, the next thing you have to do is find those little ridges. And your eyes look at them and they go, oh yeah, I know where those are, I can find them. But getting the software to recognize them was a little bit of a trick. And so I tried several different things, various averaging, median filtering, all these sorts of things to try to get to that. And now that I finally kind of figured out how to get them, it makes sense. And it's this remove outliers feature that actually ended up doing the most. And if you think about it, what's going on? The problem is that you have, as you look through this region, you have regions that are very bright and regions that are very dark, even within one of these bright colored ridges. And so by kind of going into the region and eliminating the outliers, basically, we brought those ridges down to the point where they all behaved and act like the ridges that we needed. And once they were there, then we could begin analyzing. So we actually, and this kind of jumps a little bit, I used the Fine Ridges algorithm from Bob Doherty. And basically, we did it on 3 different images, or 3 different implementations. First of all, we looked at the bright ridges. You can see the bright regions. then we looked at the dark regions, but then we looked at the transitions between light and dark. So we created, and that's the example that I showed here because it illustrates it the best. We take our ridge image, and it's not actually this image, it's this image that's been converted into, it's had this run against it. We create this variance filter, and this is essentially the slopes between the bright and the dark regions. And once we have the new bright and dark regions, we can run the fine ridges algorithm, and we create, and if you look at where all the little yellow ones are, they create boundaries between the light and the dark regions. We take those boundaries and combine them with all the other ridges, the ridges that we find from the bright regions and the ridges that we find from the dark regions. And now we do an okay job. It's not perfect, and that's one of the things that I'd like to keep on working on it. But anyhow, we find all these ridges, and then the next thing we do is we quantify them in terms of how they correspond to the pattern that we care about. And if you look around, sometimes this little blue ridge right here, this one we don't really care about. And what we really care about as we look at it is the ridges or the features that are all pointing toward the center of the image. And so what we ended up having to do was filter these guys out based on whether or not they were pointing toward the center of the image. And the lines are relatively straightforward. You calculate the slope of the line. If the line intercepts with the center of the image, then it's a keeper. If the line goes off in some weird direction, then you throw it away. The chevrons are a little bit trickier because you want to make sure that the point of the chevron is aimed at the center of the image. And that's where things got a little more interesting, because some of these chevrons are very well shaped. You can see that. That one's pretty straightforward. But then, let's see if I can find it again. This guy is definitely a chevron, but his tail comes way out here. So the next step was to come up with a way of identifying how these features were oriented. So what I did is I went into the list of pixels around the edges, the boundary pixels, found the two ends of the feature found the two ends of the feature, and then figured out which end was closest to the center to decide whether or not the feature was actually pointing toward the center, and that was fine. For the chevrons, and it's easiest, let's see if I can do this without shaking too much. For the chevrons, it's easiest to treat them like a triangle. And the base of the triangle are these two outer pieces, the two outer ends. And then the peak of the triangle is the point that's furthest away from the line defined by two ends. And so what we do is we take the midpoint of the base as a vector up to the peak. We ask the question, is that line pointing at the center of the image? And if it's pointing at the center of the image, then we keep it, and it would be color-coded in yellow here. And if it's not pointing toward the center of the image, it stays in a different color. And so, in fact, so the ones that come from the variance image are here. The ones that come as bright ridges are in the light blue. Dark ridges are in the dark blue. All right, to show that it actually works, what happens is you go in and you start doing this algorithm, and you discover that there's a whole lot of sort of coherent or consistent pixels that can become possible candidates for being one of these ridges. But you can see that by and large, these guys are all pretty randomly oriented, and only the yellow ones are kept. And so this is the tally of the number of lines, the number of chevrons, the summed line length, and then this ridge number, which is a combination of the contrast of the ridges combined with the lengths of the ridges. Anyhow, you can see that we actually do a reasonable job with them. So then the next step is to correlate the ranking from the observers with our predicted value. We see that they actually come fairly close. This was done in the JMP software, and it was a principal components analysis. We felt pretty good about the numbers. We'd like them to be a little bit closer, but we are working against panel rankings. So to summarize, summarizing, The image J functions we used were the remove outliers, the variants, the find ridges from Bob Doherty. Then we also used Gabriele Landini's morphology package to get the binary connectivity, and then managed everything with that. The skeletons, as I described, were found or were decided whether or not they were pointing at the center, whether we were keeping them. The pointing direction was determined by treating the chevron as a triangle and deciding if it was pointing at the center. Pretty good correlation. We obviously have some room for improvement if you're looking critically at any of those images. And that's basically it. Now, this is the standard Dow Chemical legal disclaimer. I'm not going to read it. Then the last thing I want to point out, and I would love to have all of you guys submit papers, we're going to be organizing a session at the Microscopy and Microanalysis 2016, specifically about image processing and other things related to imaging, including their display, the analysis, and that sort of thing. So I'm sure all of you who are members of the Microscopy Society will be getting notices, but I wanted to pre-announce it, get you guys thinking about what you might want to do. Thank you for your time.